Orin, is this okay for the screen?
Yes. This one is fine.
So I'm all set.
Yes.
Great.
Okay, we're going to wait a little bit more.
Sure.
Miss Elisa, can we start now? Wait for a second, I need to check.
My clock is still 2 minutes.
Okay.
Yes, Bu Purin, but don't you need to wait for the head of the department?
Oh, yeah.
Oh, yeah, she's here now.
This year.
Yeah, she's here.
So are you taking care of the recording, starting the recording?
Yeah, we already started.
Oh, it's already started. Okay.
Yeah. So you are in the third floor now?
Yeah. In my old room. It's very clean.
It's very clear. No one moved in your old room?
Not yet.
Not yet.
And they cleaned it, so it's really clean.
You look it better?
I sent also the
PDF.
presentation to you in WhatsApp.
Yeah, I already have it.
Okay. But probably everybody can also download it, right?
Yes, I can send it to the group now.
Okay.
Oh, okay. I think it's about the time to start. So good afternoon, everyone. Welcome to today's talk on transcriptional regulation of monoterpenoid indole alkaloid biosynthesis in Madagascar Periwinkle. And my name is Purin Chandrapurnama, and I'll be your moderator for the next 1 hour. So before we start, let me quickly go over today's agenda. The first session is opening, and then the next session will be introduction, and the third section is presentation. The fourth session is opinion question answer session, and the last session is closing. Well, to start, please welcome Dr. Santi Listiawati as the head of department to open our today talk. Please, Ms. Santi.
Thanks, Ms. Purin. Good afternoon, ladies and gentlemen. Thank you to each and every one of you for being here today at this wonderful moment. My name is Santi. It's an honor for me to speak on behalf of the biology department. Let me begin by giving a warm welcome to the lecture on transcriptional regulation on monoterpenoid indole alkaloid biosynthesis in Madagascar Periwinkle. I'm very thankful to you, Professor Johan Memelink, the head of Plant Physiology Laboratory, Institute of Biology, Leiden University. Thankful for taking out of your busy schedule to attend and speak in this event. Before we begin this lecture, I would like to express my heartfelt gratitude to all of you who have sincerely committed to this event to make a success. This event would have been impossible without the support of each and everyone who are present here. I hope that through this event, we all can learn new things, especially about transcription regulation, secondary metabolic in Madagascar periwinkle. I wouldn't like to take too much of your time, so I will hand it over to our moderator to process the next agenda. Once again, welcome and thank you very much for attending this event. Ms. Purin, please. Unmute Ms. Purin.
I'm sorry. Oh, thank you very much, Dr. Sati, for opening the talk. Now let me introduce our guest speaker for today, Professor Johan Memelink. He's the leader of Plant Cell Physiology Lab, which mainly work with this topic. And he's here today to share his expertise with us. Without further ado, I'll hand it over to Johan for the presentation. Please, Johan, you can start.
Okay, good afternoon, everybody. Here in Leiden, it's 10 o'clock in the morning, so a little bit earlier, but not too early. Okay, so I want to thank you for the invitation to talk about some of my work. And as already indicated, I spent quite some years on studying the monoterpenoid indole alkaloid biosynthesis pathway in the medicinal plant Madagascar periwinkle. And on the left of the picture, you see a photograph of the plant. So it's a very nice ornamental plant, which is which I have no idea whether it's common actually in Indonesia, but here in subtropical regions, so south of France and the south of Spain and Portugal, you see quite a lot of this plant as an ornamental plant. It has very nice flowers. But actually, I do not work with plants per se, but I work with cell suspensions. That's the picture that you see on the right. So it's these are suspensions of unorganized cells, which grow in Erlenmeyer flasks and which are in our regime transferred weekly. And yeah, the advantage is that you are working with a source of cell material which is uniform and which is available every week. So you do not have to sow plants, you always have this culture available. And this culture has been quite useful actually in our studies. So all our studies, almost all our studies, I should say, were done with these cell suspensions. Okay, let me see, yeah. Well, as all of you are aware, plants are characterized by the fact that they produce so-called secondary metabolites. So every plant species has a certain spectrum of secondary metabolites that it produces. These secondary metabolites are defined as small organic compounds with molecular weights of around 1 kilodalton. They are also called sometimes specialized metabolites. So they are distinct from the metabolites that form part of the primary metabolism that's necessary for cell growth, plant growth. Sorry, is there a poem?
No, you can proceed.
Okay. Their role in plants is in communication with other organisms. So many of these compounds are involved in defense, but these compounds also, certain members of these group of compounds also have roles in communication with positively acting organisms. And my research, of course, I should mention maybe that these secondary metabolites are also of interest because many of our common day pharmaceuticals are derived from plant secondary metabolites. So about 30% of common of current medicines are actually plant secondary metabolites or derived from plant secondary metabolites. Okay, so my research interests have been to find the genes that encode enzymes that are involved in secondary metabolism. And I was especially interested in the regulation of these genes by transcription factors. So this research involves expression of these enzymes, looking at the expression of enzymes at the gene and protein level, and at their regulation by transcription factors. And the idea with the transcription factors is actually that if the system would be such that the whole pathway is regulated by one or a few transcription factors, then these transcription factors could also be used to improve the expression of the pathway and the production of the compounds. Okay, so these monoterpenoid indole alkaloids are produced by this plant, the Madagascar periwinkle. The Latin name is Catharanthus roseus. Its former Latin name is Vinca rosea. This has been changed a couple of decades ago. And shown here are a couple of compounds that are used as pharmaceuticals. So in the top, you see esmelicin and serpentin, which are used against high blood pressure. And at the bottom, you see two anti-cancer alkaloids. And these alkaloids are called monoterpenoid indole alkaloids because they have dual biosynthetic origin. So they contain a terpenoid moiety, a monoterpenoid moiety, which is indicated in blue. And they have an indole ring, which is derived from the amino acid tryptophan, which is shown in yellow. And these two compounds are so-called monomeric monoterpenoid indole alkaloids, and the anti-cancer compounds are dimeric monoterpenoid indole alkaloids. So there are two of these monomers coupled together to each other. So especially these two anti-cancer compounds are very important pharmaceuticals from this plant. They are currently still commonly used in fighting cancer. And these are, at the bottom, we see the natural compounds, vincristine and vinblastine, with their various trade names. So it depends on the producer, what kind of trade name is used. So many pharmaceutical companies sell these compounds. These compounds are now generic anti-cancer compounds, so the patents have run out, so anybody can produce and sell them. But some companies make modified derivatives of these natural compounds. So we see here two compounds, vinflunin and vinorelbin, which have modifications here at the top indicated in red. And these modifications are made to try to improve the properties of these anti-cancer compounds. So, for example, the solubility or the toxicity or uptake, these kinds of parameters can be changed maybe by these modifications. And then these modified compounds are again tested in clinical trials. And then if they have improved properties, they are available again on the market. So for example, these two compounds are now available on the market and are claimed to have better properties than the original natural compounds. And the way they work as anti-cancer compounds is that they block microtubule assembly. So microtubules are very important for cell division. And since cancer cells are the most actively dividing cells, these compounds preferably inhibit cell division of cancer cells. Okay, so this is a schematic overview of the pathway. In total, the pathway counts about 45 enzymatic steps, which by now have all been resolved. A number of these steps have been resolved by us in Leiden, but many other steps have been resolved by other groups quite recently. But the whole pathway is now known at the enzymatic level. This is not the case for the transcriptional level yet. So let me just go through a few details of the pathway. So the indole ring of these alkaloids is derived from the amino acid tryptophan, which is converted into tryptamine by one decarboxylation step by this enzyme TDC. And then the monoterpenoid part is derived from the primary metabolite geraniol, and then in a number of steps, this compound is made cyclogonin, and these two compounds are coupled together by the enzyme strictosidine synthase to form the first monoterpenoid indole alkaloid precursor, strictosidine. And from this precursor, various end products that you can find in the Madagascar periwinkle plants are formed. And in addition, from some of these compounds, the dimeric alkaloids are made. So what I indicated here is two parts of this pathway, which I will talk about a little bit more in the rest of my talk. So this first part of the pathway to produce this monoterpenoid part is called the secoiridoid pathway. And then this part of the pathway where strictosidine is formed and the first steps from strictosidine onwards, I will indicate sometimes as the middle part of the pathway. So this middle part includes, among others, the enzymes TDC, STR, and this SGD. So what I indicated here in yellow are a few enzymes that we discovered in Leiden, and this was in the 90s. So between 1990 and around 2000. So it's already 25 years ago. So these are the genes that we worked in those early days. And when we were looking at the expression of these genes in the cell suspensions, we noticed that the expression of these genes can be turned on by the plant's defense hormone, jasmonic acid, or what we are actually using commonly is methyl jasmonic acid because it's cheaper. So what was done here is to treat cell suspension with a certain amount of this methyl jasmonic acid, and then samples were collected over time in hours indicated here. And then in the RNA, the expression level of the different genes was measured by northern blotting. So essentially the bands that you see are the messenger RNA bands, and the more you see of these bands, the more there is actually, so the more transcript there is. So you see that for these 5 genes, when you induce with jasmonic acid, you have an increased expression of all these genes. So as a loading control to show that we have identical amounts of RNA on these gels, we use this control gene. So this is a household gene. So this shows that methylgesmonic acid or gesmonates have a strong effect on the expression of these genes, and this I found very interesting, because it shows that with this compound you can upregulate at least all the genes that we had at the time. So I was interested how this was regulated at the transcriptional level, so which transcription factors are involved in establishing this increased expression of these biosynthesis genes. And well, this is just a kind of summary of around 15 to 20 years of work. So we found a number of transcription factors. First, we found the ORCA transcription factors, which are activating transcription factors, which regulate genes in the middle part of the pathway, for example, the TDC and STR genes. And these ORCA transcription factors are regulated by another transcription factor called MYC2. CR stands for Catharandus roseus, MYC2. And this MYC2 transcription factor is normally actually repressed by a family of repressors, which are called JAS. And I will explain this model a little bit later in a few slides later. But just to show the importance of, for example, these transcription factors. So here we look at the expression of the different transcription factors and also the biosynthesis genes after, again, treatment of the cell suspension with methylgesmonic acid for half an hour or 6 hours. And then we see that all of the genes that we are looking at are expressed at a higher level when you treat methylgesmonic acid. So it's not only the biosynthesis genes, but also the transcription factor genes respond to methylgesmonic acid. So this is actually the way that these biosynthesis genes can respond to methylgesmonic acid, because the transcription factors themselves respond to gesmonic acid. What you can also see is that the transcription factors respond much earlier than the biosynthesis genes. So first you switch on the transcription factors and then you switch on the biosynthesis genes. And what we did here is actually an experiment to show that methylgesmonate and these transcription factors are very important in regulating the biosynthesis pathways. So it was found out that if you delete a very small part at the C-terminal end of these JAS repressor proteins, that you can turn them in so-called dominant negative forms, which cannot be switched off anymore by gesmonic acid. So we used this given as to produce cell lines that overexpress this dominant negative form in a fusion to GFP, the green fluorescent proteins. This is just to visualize the proteins. And we are looking here at expression of genes in 2 different cell lines. So we have a control cell line, which is transformed with the GFP gene. And we have a cell line overexpressing this GFP JAS, dominant negative JAS fusion. And what we're looking here in the 2 top panels is at the expression of the transgenes that we introduced. So here we're looking at the expression of the GFP gene. So here the GFP band is a little bit larger because GFP is fused to JAS. This is just a gel electrophoresis of the RNA. So the position of the RNA also indicates the size. And what you're looking at here is the overexpression of this fusion gene. So it's highly overexpressed. What you're looking at here is at the expression of the endogenous JAS1 gene. So you're looking here, what you're seeing here is induction by jasmonic acid. So you see that's slightly induced, but the expression is very low compared to this transgene. And now the interesting thing is that if you look at all the downstream genes, the transcription factors and the biosynthesis genes, then in this cell line expressing this dominant negative form, the expression is very strongly turned down. So that means that all of these genes are under control of these repressor genes. Even the JAS genes themselves. So we're looking here at the endogenous JAS genes. They are also controlled by these, auto-controlled, I should say, by these repressor genes. So to go back again to this model that we have, so what happens is that the JAS repressors are normally expressed in the absence of jasmonic acid to a certain level, which is high enough to repress this transcription factor. This repression is indicated by this symbol. This is negative and this is positive. And when plants produce jasmonic acid, so the active form is jasmonate isoleucine, then this is perceived by the receptor, COI1, and this leads to degradation of these repressor proteins and activation of this transcription factor CMYC2, which regulates these other activator transcription factors, ORCA2 and ORCA3, which regulate the middle steps in the monoterpenoid indole alkaloid biosynthesis, including the TDC and STR genes. So this is this model summarizes things that we discovered between, say, 1995 and 2010, 2011 maybe. So this is also already a while ago. But I want to talk a little bit more about a thing that we did more recently as part of a larger project. It's a European project, which was called the Smart Cell Project. So it was an EU project running from 2008 to 2013. So this involves quite a large amount of money. And it has to be a joint effort. So we had 14 academic partners from different EU countries. And in addition, we had 3 commercial companies that were participating. And the title of the project was the elucidation and metabolic engineering of the seco-iridoid pathway in Catharanthus roseus. And that was because the seco-iridoid pathway at that time, so this proposal was submitted to the EU in around 2007 or so. The seco-iridoid pathway was a black box, so the enzymes that were involved were unknown. And we thought, okay, let's try to solve this pathway with this joint effort. So this shows the situation before the Smart Cell project started. So these genes were known. So we had isolated ourselves this G8O gene. Yeah, which is the gene that we had here in this expression analysis also. And these genes were also known, the LEMT and SLS gene. They were already isolated by other groups, but what was in between was a black box. So these iridoids or seco-iridoids are not only interesting for Catharanthus roseus, so they occur in many plant species. So I've listed a few compounds from different plant species where they accumulate as end products. So in Catharanthus roseus, secalogenin is an intermediate to the formation of these monoterpenoid indole alkaloids. but in some plant species, large amounts of the stegoiridoids accumulate as end products. So, for example, in this plant, which apparently is from Japan, Acuba japonica, which is a common garden plant in the Netherlands. It accumulates large amounts of alkubin. And in this plant, Plantago, which is also a common European plant, this compound accumulates. Then in catnip, which is also a garden plant actually in Europe, this compound is accumulating, which apparently cats do not like. So it chases away cats. And in well, different other plants you can find cyclogonin. So this is Lonicera, and this is the white snowberry, the Symphoricarpus albus. These are all common European plants. Okay, so the idea of the Smart Cell project was to generate a database, a sequence databases of Catharanthus roseus. They did not exist at the time. So all the work that we had done until then was without having any extensive DNA databases. And to look in these databases for genes of interest by various means. And then when you identify genes of interest, you can then try to functionally characterize these enzymes and confirm that they are involved in the biosynthesis of these monoterpenoid indole alkaloids, either by expressing them in a heterologous system, for example, in E. coli or in yeast, and then doing an in vitro enzyme assay, or expressing them in plants. So try to characterize them in planta. And what we used as a system is overexpression by agar infiltration. I will talk a little bit later about it. And then hopefully you can determine the function of these genes of interest and determine whether they have a role in the alkaloid biosynthesis. So that was the idea. And for finding candidate genes, we used a very common strategy, which is co-expression analysis strategy. So this is used, has been used in plants for finding many candidate genes successfully in various systems. And it works as indicated here. So the assumption is that genes in a metabolic pathway have a common regulation, so that all or a group of genes in the pathway have the same type of regulation. And what you need to perform the strategy is you need at least one known gene in the pathway. And then you need conditions that affect the expression of this gene in a special manner. And then when you have these two things, you can look at other genes that have the same expression pattern and that might be candidates for a certain enzymatic step. Okay, so we had two known genes. So we had, this is actually a precursor gene to make geraniol, which is not really part of the stegoiridoid pathway. And we had this first gene, geranyl 8 oxidase. So this was perfect. And then we knew that the defense hormone jasmonic acid induces these genes. So that's one condition that you can assume is common for all the biosynthesis genes. And you can look at tissue-specific expression. I will explain this in the next slide. So when you look at the biosynthetic pathway, there is another type of schematic overview. This overview is based on work from a French group, which was done between 2000 and 2010, where they looked at tissue-specific expression of different genes. And it turned out that the secoiridoid pathway was expressed in a certain cell type called the internal phloem-associated parenchyma. So this is a cell type in leaves. Whereas the middle pathway, including TDC and STR, was expressed in the leaf epidermis. And this pathway leading to vindelin, which is a part of the dimeric alkaloids, this branch is expressed in lenticular and idioblast cells. And this shows a picture of this expression. So this is results from an in situ RNA hybridizations from this French group, where they looked at the localization of gene expression in the leaf. And so this is a Catharanthus leaf section, what you see here. And this is looking at the expression of G8O, which is expressed in these patches of cells, which are the internal phloem-associated parenchyma cells. And then here we have the TDC gene, which is, so this, what you see looking at here is this last part of the leaf, which is expressed in the epidermis, so the outer cell layer. And here we have a gene from the vindelin branch, which is expressed in these cell types that you find inside, that are scattered like inside in the leaf. And these are the lenticular and idioblast cells. So we have here 3 types of cell-specific expression in the leaf. And this was used, among others, to identify candidates. Okay, so the strategy of the smart cell proposal was to deduce the most likely pathway from geraniol to secologonin. So this was just sitting down behind your desk and looking at the structures and looking at, okay, which type of enzymatic conversions would be necessary to convert this structure to the next structure and so on. So then you kind of point out which kind of enzymes are likely to be involved. And then you can, usually these enzymes have a certain signature, so you can recognize these enzymes by certain motifs. So you can look in the database for enzymes that have these kind of motifs. So that's how you can find candidates genes from a transcriptomic database. And then when you have these candidates, you can try to express them in a heterologous system and purify the enzymes and look at which kind of enzymatic reaction they catalyze. So that's what we did. So we used here both E. coli as well as yeast for as heterologous expression systems. And so this is the pathway that we came up with on paper. So in black are the genes already known, so G8O and LAMT and SLS. And then we came up with these unknown genes. So there's one gene that's, or one enzyme that can make this compound, so it can oxidize here and here. So there's 2 OH groups which can be oxidized to a double bonded O. So this can go either like this or like this, first the one and then the other, to have this dialdehyde, and then you have this conversion, and then you have still a few steps to get this LAMT. So in total, we thought that there would be 1, 2, 3, 4, 5 enzymes missing that we were looking for. So we established transcriptomic databases. So this was mainly part, this work package was mainly done by the Belgian partner from the Research Institute VIB in Ghent. Although we from Leiden supplied these RNAs, these are all cell suspension RNAs, and these RNAs were supplied by the Finnish group. So our RNAs were from cell suspension treated with methyl jasmonate, and from two cell suspension that overexpressed the ORCA transcription factors, which regulate the middle pathway genes. This is with the idea that, okay, these transcription factors could also regulate a part of the saccharoid pathway. So this could be a condition that regulates these genes and could identify candidate genes. And the Finnish RNAs were plant RNAs that from plants that were treated with methyl jasmonate for various times. Okay, so the transcriptomics analysis led to a large amount of data, of course, as usually with transcriptomics. In total, more than 36,000 contigs, so essentially genes were found, which is kind of the type of number that you usually find with these genome-wide transcriptomic analysis. So this led to, say, these 32,000 almost genes. And these genes were deposited in a database, the ORCA-A database. I didn't check it recently, but if all is well, this database should still exist. And then this database was mined for candidates missing iridoid genes. And we also looked at transcription factors that might regulate these genes. I will also talk about this a little bit later. And what I will not talk about, but which would be interesting, is also transporters that transport these iridoids, because the fact that different cell types in the leaf are involved in the biosynthesis apparently also indicates that transport in the leaf is an important factor. But I will not further talk about this now. Okay, so this is an example from a gene that we found from co-expression analysis. So this is what it looks like. So this is the way of representing the expression level. So in blue indicates high expression, and yellow low expression. So this is false color coded. And we are looking here at 4 genes. So we have the STR gene from the middle pathway. And we have here the 2 known genes from the saccharoid pathway. So the precursor gene and the first gene from the saccharoid pathway. And then we found a candidate gene. It was a so-called alcohol dehydrogenase gene, ADH10, with this expression pattern. And when you look at this, you see that this expression pattern is very similar to the G8O and GES genes, and very different from the STR gene. So this was a candidate gene for the HGO step. And then I include here a statement, and that's because we had a problem with this in our smart cell project also. So we use this very fancy novel omics technique. So we use transcriptomics and metabolomics. But when we made our proposal, we forgot that to characterize the enzymes, you actually need classic chemistry and enzymology. So for example, for determining the most likely pathway, you really need classic chemistry. So you need an organic chemist who can tell you, okay, this is the most likely pathway. And then to test the enzymes, you just need classic enzymology. So you cannot you cannot forget about these classic techniques when you use these novel techniques, and that's what we did a little bit in our proposal. And during the project, we discovered, okay, we need actually a number of things that we did not think about when we proposed this large proposal. And one of the things that we did not think about was to make the standards for the enzymatic assays. So to validate these candidate enzymes, you want to do an enzymatic assay. So you overexpress the enzyme in a heterologous system, and then you purify it, or you have an appropriate control, and then you do the enzymatic reaction. And to do the enzymatic reaction, you need the substrate. And if you want to determine that the correct product is made, you also need the product as a standard. And then you run into the problem that you cannot buy all these chemicals from the Stigma catalog or from any catalog, so they are not commercially available. And so that means that we had to find ways to get these compounds. And so my PhD student actually looked a little bit in old literature. So he looked actually in literature from the 70s. So it's really looking back in time. And he found a group that at that time did bioorganic synthesis. And he found out that when you start with this natural compound, ocubin, you can make via relatively simple organic reactions the different intermediates. So we looked for a chemical company, and we found one in Holland that was willing to do this kind of biosynthesis. And they told us, okay, we can make 100 milligrams of these compounds if you give us 10 grams of ocubin. And ocubin is also a compound that you cannot buy. So my PhD students, or actually together with my PhD student, we went one morning to this building, which is in our campus. So we went there on Saturday at 7 o'clock, and we hid behind this bush, which is an Aucuba japonica bush, and we harvested a large amount of leaves. So we took 2 shopping bags and filled them with these leaves. Well, actually, the shrub is really large. So when we were finished and had harvested 2 large shopping bags of leaves, we could still not see that anything was missing from the shrub. But anyway, so my PhD student went back to the lab, and with these 10 kilos of Aucuba japonica leaves, and he isolated via this protocol that he found, which dated from the 70s, this ocubin. It was a relatively simple charcoal absorption and silica gel chromatography purification protocol, and then the purity was checked by NMR, and indeed we had exactly 10 grams of this ocubin. And then we sent this ocubin to this company, which is localized in Holland, Chiralix, and they synthesized the different compounds via the organic synthesis schemes that we gave them, and they delivered these compounds to us as glucosides. These are the stable forms of these compounds, and then if you need the deglycosylated forms, you can just deglycosylate them just before use with a general deglycosylase. So these are not, the deglycosylated forms are not stable, so you have to do this just before use. So this all worked perfectly. So we obtained very nice standards from Chiralix of the different intermediates. So here we have again this 8-HGO gene, that's candidate that we discovered via this co-expression analysis. And here is the enzymatic analysis. So the top 4 chromatography runs are the standards, so that were made by the Carelix company. And this is the reaction, the run of the reaction. So here, 8-hydroxygeranol, the substrate, which is the peak number 1. No, it's not. It's peak number 2. Exactly, it's peak number 2. Was incubated with the purified HGO candidates, and the cofactor is NAD+. So it's a reduction reaction or oxidation reaction, and indeed the different products that you expect are formed. So you find the products that have one of the two hydroxy groups oxidated, or both, just as expected. And here is to show that we did the MS spectra to show that the different peaks were identical to the chemical standards, which they were. So we did this for all the different enzymes candidates that we had, and this resulted actually in the discovery of all the different enzymes. So this was quite successful. And we also looked at the tissue-specific expression of the different genes that we discovered. So this is the gene that we already had, and these are new genes. So this is the HGO gene that I just talked about so much, and these are the next genes in the pathway. And you see that they are all expressed in these IPEP cells. So the whole sequence of genes is expressed in this specific tissue type. Yeah, this is a phylogenetic tree of HGO and other alcohol dehydrogenases, and I just show this to show that in this tree are several alcohol dehydrogenases from Catharanthus. So here indicated with the red dots. So here are 2. And here's another one, and here's one that's actually in the database from NCBI was labeled as 8-HGO. And we think it's very likely, we didn't test this, but it's very likely that all of these enzymes can catalyze this, this step. But we don't think these are the real HGO genes, because this one is expressed actually in the IPEP cells, whereas all these genes are not. So probably these can perform the reaction, but they are expressed in the wrong tissue. So by a combination of the enzymatic reaction and the tissue-specific expression, we think that this is the correct candidate. Okay, so this is the overview from at the end of the Smart Cell project. Which was in this respect quite successful. So we did what we wanted to do, is to elucidate the complete saccharoid pathway. So these are the novel genes of the known genes. Here is indicated in double because this was already identified by another group, but we also identified this as a candidate in our Smart Cell consortium. The same here for this gene. This was actually while we were busy with our work, another group published this IS enzyme, and we already had this in our collection of candidate enzymes. So for this enzyme, we were scooped, and actually just this enzyme was published in Nature. And we also published the whole pathway in Nature. But so we have here a number of new enzymes, so the HGO and these 4 new enzymes, and this one, yeah, we were scooped, but in principle it is also a new enzyme to form this sacchalogonin part of the pathway. Okay, so to show that these genes actually could or were sufficient to produce from geraniol cyclogonin, we used an implanta assay, and this is called the agro-infiltration assay. And the way this works is that you introduce the genes in Agrobacterium tumefaciens, the well-known plant genetic engineering bacterium. And as you probably all know, this Agrobacterium can transfer part of its tDNA plasmid to the plant, where it gets expressed. And the nice thing about this agro-infiltration is that you can mix different Agrobacterium strains with different genes. So you do not have to introduce all your genes in one plasmid in Agrobacterium, but you can put every gene on a single plasmid, make an Agrobacterium strain, and then make a mixture of the different strain with different genes, and then co-infiltrate, as it's called, leaves with this mixture. The co-infiltration is done with a simple syringe. So you just kind of press the liquid culture on the leaf and thereby infiltrate the spaces between the leaves with the mixture. And then you incubate it for 5 days. And what happens is that the Agrobacterium transfers the genes to the plant cells where the genes are expressed. And then you collect after 5 days these leaves, and you can do, for example, an enzyme analysis or a metabolite analysis with these leaf parts. So that's what we did. So we mixed the 12 genes that are in this scheme, and we agro-infiltrated them in this Nicotiana benthamiana. You may ask why Nicotiana benthamiana. So this is the commonly used plants that kind of everybody that does this kind of assay is using. And the reason is it grows fast and it's easy to agro-infiltrate. That's really the only reason that this plant is chosen. And so we were successful because with this combination of 12 genes, we were able to make from geraniol, or actually from these two precursor compounds, the compound strictozidin. So this is described in this Nature paper. So indeed, these genes are sufficient to produce the cyclogonin, which is necessary for producing the strictozidin. Okay, so this was very nice. So then this was actually when the Smart Cell project was already officially finished. But yeah, you have all these data and still PhD students that are looking for work. So we continued a little bit with all the data that we had. And we discovered a transcription factor, BIS1, which had exactly the same expression as the iridoid pathway genes. So again, we are looking here at different types of tissue. So these are the tesmonate treated cell suspension. These are the ORCA overexpressing cell suspensions. And then yellow is again high expression, blue is low expression. And here are the middle pathway genes like STR and the ORCA genes. And these are late pathway genes. And here at the bottom are the sacroiridoid pathway genes that we just discovered. So you see here G8O and 8HGO and so on, IS. And this BIS1 transcription factor has exactly the same expression pattern as these iridoid genes. So we thought, okay, maybe BIS1 is responsible for regulating these genes. So what we did in Leiden is we overexpressed BIS1 in cell suspensions. And that's what you see here. So we have here control cell suspension. actually we have 3 control cell suspensions. So each lane is an independent cell suspension, and we have 3 overexpressing cell suspensions. And so this is the BIS overexpression. And this is an analysis of the expression level of the different genes. And what you see is that in these BIS overexpressing cell lines, all of the genes from the MEP pathway, which is the precursor pathway for the sacroiridoids, and the iridoid pathway is overexpressed. So you see a very strong expression of all these genes. And there's no effect on the middle pathway. So you see here the TDC and the STR gene, for example, overexpression of this transcription factor has no effect on the expression level of these genes. So this indicates that the BIS1 transcription factor very specifically regulates actually the whole precursor pathway and the iridoid pathway. And then also the alkaloid production was analyzed in these cell lines, and this was work that Purin Purnama also contributed to when she was in Leiden. So this was around 2014. And so what you're looking here at is at the levels of the different intermediates, so the iridoids and the alkaloids that were found. These are also alkaloids that were found in these cell lines. What is missing here is the control cell lines, so we do not see any bars here for the control cell lines. And that's because all these compounds were not detectable in the control cell lines. So essentially, I cannot say that the level was 0, but it was below the detection level. But in the transgenic overexpressing, BIS1 overexpressing cell lines, we found well detectable levels of the sacroiridoids and of streptozidin and of other alkaloids. So this shows that BIS1 has overexpression has a very strong effect on the expression of the iridoid pathway genes and on the production of alkaloids. So this is to summarize the findings, including the findings from the Smart Cell project. So we have here, we have characterized during the Smart Cell project all the genes that are indicated here in red that lead from the from geraniol to the production of sacologonin. And also indicated here are the genes that were not characterized by us, but by others from the MEP pathway and from, this is all the MEP pathway. And all these genes in blue turn out to be regulated by BIS1 and a related transcription factor BIS2, which were also characterized in the Smart Cell projects. And then you have below the genes in the middle pathway, which previously we had already shown that they were regulated by the ORCA transcription factors. So we have, regarding the transcription factors now, a relatively complete picture about which transcription factors regulate these parts of the pathway. So this picture does not is not complete for more downstream parts of the pathway. So the transcription, so all the enzymatic parts steps are known by now, but the transcription factors are still unknown. And also it indicates that you cannot upregulate the pathway by overexpressing just a single transcription factor, but you would need multiple transcription factors. So separate transcription factors for the iridoid part, transcription factors for the middle part, and unknown transcription factors for the more downstream part. So it's a quite complicated type of regulation. So the question is whether transcription factors are really useful to influence the expression level of this pathway, because it's so complicated. But okay, on that note, I want to finish with pointing out all the people that were involved in the Smart Cell projects, at least. So from Leiden, we had a number of people. So the staff members were myself and Rob Verpoorten, and the PhD student that did here all the work, who was paid by Smart Cell, was Karel Mietinen. And Purin Purnama was here as a visiting scientist, and she contributed to the metabolic analysis. And these are bachelor and master students that were involved in the project and were quite successful in this project actually. And these are members of the other teams that contributed to the final results. And with this, I would like to thank you for your attention, and I'm open to questions if there are any.
Yeah, thank you very much, Johan, for your insightful presentation. I'm sure that our audience has learned a lot from you today. Before we move on to the question answer session, I have a few questions for you. Is that okay?
Yes.
Okay. First of all, how did you become interested in this topic? And what motivates you to pursue this field of study and research?
The reason is a little bit simple because when I was appointed as a staff member, it was specifically on this topic. So in say around 1990, when I was appointed, Leiden had a collaboration project with Delft. So it was called the Biotechnology Delft Leiden project, where Leiden was looking for metabolites and genes, and Delft was looking into bioreactors and methods to culture cell cultures at large scale. And the idea was to use genes to modify biosynthesis in cell cultures and then grow these cell cultures in large bioreactors. And so my appointment was to look really at the genes and at regulation in Caterantes, and that's how I became interested in it.
Okay, next question. What are some of the practical implication and application of your research? How can it benefit the society and industry?
Yeah, ideally you would say, of course, you have a lot of knowledge about the biosynthetic steps. So essentially these steps in, for example, the saccharoid steps will be similar in all plants that produce certain saccharoids as an end product. So you know that an enzyme with similarity to the enzymes that we have found will be involved. So it helps identifying the biosynthetic pathway in these other plants, if anybody is interested. Well, specifically for the anti-cancer compounds, the answer is a little bit more complicated, because the problem with pharmaceutical companies is that they want to earn money, and they're not interested in essentially in helping humanity or society. They are interested in making money. So essentially they are not interested in having generic medicines, for example, because they cannot make money with it. And they are not interested in having a very efficient, highly producing plant cell line, because then anybody could produce it for low cost. They want something that's actually so complicated they can only do it so that they can sell the product. So that's a little bit the problem with the way our society is organized at the moment, that you can provide a lot of knowledge, but at least via pharmaceutical companies, this will not lead to any advantage for society.
Yeah, thank you. The last question. Do you have any advice or tip for our audience who wants to learn more about it or get involved in this kind of field?
Read the papers. There's many review papers also. So if you look at it in just the NCBI database and just search for this, you will find many review papers, and the original papers are very nice papers to read. So I have included in several of the slides the references, so you can easily look them up. And then, read the papers.
Okay, thank you very much. Now let's open the floor for a question from our audience. Please raise your hand or type your question in the chat box, and we'll call you in order, or you can also just
Can I find the chat box somewhere?
in the chat button.
Chat button, let me see.
Below.
Below.
Yes. You can
No.
you cannot find it?
I will find it.
Okay.
Ah, chat, yes.
Yeah.
Somehow I have the impression that there were already 5 chats.
Bu Purin, I have question.
Oh yeah. Thank you.
Okay.
Okay, Miss Elisa, please.
Professor Johan, thank you very much for your nice presentation. I enjoyed very much. I have a question. So when finding candidate genes involved in a pathway, sometimes you find that some candidate genes have redundant function, right? Have you ever experienced the same thing? And how do you finally select which genes among those that you later proceed with?
Yeah, if they're really redundant, it means that all of these genes are actually involved in the step. So take, for example, the ORCA genes. We believe that the ORCA genes are essentially really redundant genes, so that their function is essentially the same. They've probably been generated by a relatively recent duplication and did not really change their function after being duplicated. So they really have similar functions. But if you look, for example, at the HGO, which is an alcohol dehydrogenase, you can find in the Catharanthus genome, I think about 15 or so different alcohol dehydrogenases. Some are a little bit different, but quite a lot look very similar to the 8-HGO that we have characterized. But we don't think that these are real redundant genes because the expression pattern of these genes is different. So for the 8-HGO that we characterized, it's the only one that's expressed precisely in this IPEP type of cells. So I think if you have genes that you think are redundant, it's quite important to also look carefully, of course, at their function. So what's the, if it's an enzyme, what's exactly the enzymatic step they catalyze? But even if it's the same, then it's still useful to look at the cell type specific expression. And what might also be different is what you see in some cases is that one of the genes is expressed constitutively, while the other gene is, for example, expressed after being challenged with a pathogen, for example. So then one gene has acquired a specific function in defense, and one gene is has a function for normal biosynthetic levels.
Okay, thank you very much. But I have one more question.
Yeah.
Is that okay? This one is going to be a very basic question because I never work with cell suspension. So how does the, I mean, you explained before at the beginning of the presentation that cell suspension is kind of like randomized cell aggregates. And then how does gene expression in the cell suspension reflect the actual expression in the plant itself?
The way you generate these cell suspensions usually is that you take a certain piece of tissue. It can be a piece of leaf, for example, which you sterilize, you surface sterilize it and put it on synthetic medium with a certain hormone balance that causes callus growth. So then you have the growth of unorganized tissue. And then once you have a callus, you can then bring it in liquid culture. And then if you transfer it and culture it for some time, you can have a cell suspension that, so for example, our cell suspension is consists of aggregates of a few cells, not too many. Say between 1 to 10 cells are aggregates. So they're not huge clumps of cells, but relatively fine cell suspensions. But this can differ between different cell suspensions and plant types, maybe. And then what you end up with is a cell type that's different from any cell type in the plant. Yeah, so it's kind of a novel de-differentiated cell type. And so it's quite artificial. So essentially the expression that you find there cannot be really compared to any cell type in any plant. So that's in principle a disadvantage. However, for example, what we have been looking at is at the induction mainly by jasmonic acid. And in that respect, the response of the cell suspension is very similar to cells in a plant. So for example, with Arabidopsis, we have worked on jasmonate responses with plants and in cell suspension. We also have an Arabidopsis cell suspension. And there, the timing of expression in response to jasmonic acid was very similar, whether you look at cell suspension or at plant. So it depends really at what you're looking at, I think. So for our jasmonic acid studies, it was very useful to have the cell suspension. And we have also done one very interesting type of experiment. It's already 25 years ago almost, but which was only possible with this cell suspension, which was very successful actually. So you can do certain things that you cannot do with plants, but it remains an artificial system. So if you want to, you can find things, but you always will have to cross-reference it to what happens in a plant if you really want to know what's going on at the plant level.
Okay, I understand. So this is the differentiated cell. So at some point it's different, but still very informative. It depends on what you're looking at. Okay.
I use it as a tool for finding things. So essentially all the genes we found and all the transcription factors and the mechanisms were all done with cell suspension, and all these things are true for plants. So these were not artificial or something. They're really happening in plants. And the reason also we started with the cell suspension is because of the start of the project. So it was this Leiden-Delft interaction. So the idea of the whole project was, okay, we want to produce pharmaceuticals in cell suspensions, which can be grown in very large reactors. And this will produce secondary metabolites in an efficient way. And this all did not work because secondary metabolites are only produced in specialized cell types. And they are not produced anymore in de-differentiated cells. So actually the cell suspensions usually do not produce any important levels of secondary metabolites, except when you treat them, for example, with jasmonic acid, or when you overexpress certain transcription factors, you can change that to some degree. But this is the reason that almost nobody is really using cell suspension for producing actually pharmaceuticals, for example. They're still always isolated from the original plant source.
Okay, thank you for the answer.
Yeah, thank you very much, Ms. Elisa. I have many questions from students. We can start from Melita, please.
Yes, I'm sorry if I cannot turn on my camera because my bad connection, and I'm still a beginner, but I want to ask a question. What if it turns out there is a problem with the transcription factor, like there is some mutation that caused by the make suspension? How can we solve it without having to start from scratch, especially if the species we are using is in danger or threatened with extinction, so it cannot be utilized successfully? Thank you.
What kind of mutation are you talking about?
Gene mutation that caused by the make of suspension, sir.
Okay, so you are asking if you make a suspension and what if during making the suspension a mutation happens? Is that the question?
Yes, sir.
I don't know what to do. Maybe I should also say that our cell suspensions are polyploid. That's another thing that happens when you make cell suspensions. So Catharonsis cells are normally diploid, and actually the same happens with Arabidopsis cell suspension. Arabidopsis cells or plants are also diploid, and when you turn them into cell suspensions, they become polyploid. So I'm actually also not completely sure whether there is a real risk for mutation because you have so many chromosome copies already. But yeah, if you have, it's possible that you have mutations. These cell suspensions also change over time. So when you have a cell suspension in your lab and you keep it for 20-25 years, for sure it will be at the end of the 25 years completely different from the one that you started with. So that's another problem. And the way we solve that is by cryopreserving the cell suspensions. So we have our cell suspension in liquid nitrogen. And once every couple of years, we take a sample from liquid nitrogen and start over again to kind of go back to the original cell suspension. But it's a problem that you have to take into account, yeah, for sure.
Thank you, sir, for the answer.
Next student, maybe? Fatihah Tunisa. Nisa is not there?
Yeah, okay. I want to ask about, as you can see, a secondary metabolic biosynthesis in Catharanthus roseus is controlled by transcription factors. But one transcription factor does not only regulate metabolic synthesis through one pathway or one component, and also there are other transcription factors that also play a role. So how do we know which transcription factors that play the most important role, or they act like as a main regulators?
Yeah, that's a very good question. It's true that any gene will be regulated by a whole range of transcription factors. And each of these transcription factors will probably have a major role in some aspect of the expression of the gene. So what we were focusing on really is on the jasmonate response when we were looking for transcription factors. And the way we came quite specifically to this point is that our approach was to look for DNA regions which would allow a gene to respond to jasmonic acid. So it's a part of the promoter. So we made mutations, and in this way we localized a very small DNA region that was, when you mutated, would abolish the ability of the gene to respond to jasmonic acid. And when you would just take this element and fuse it to another gene, it would allow that gene to respond to jasmonic acid. So we had like almost 10 base pairs region that was responsible for the response. And with this, we looked for the transcription factor by essentially just binding to this region in a very specific way. So we also had mutants of this region. So the transcription factor should not bind to the mutants. And in this way, we were able to find specifically the ORCA transcription factors. But it's true that if you're just looking for transcription factors that bind to the promoter, you will find dozens or hundreds of transcription factors. And then you have no idea, in principle, in what aspect of the regulation they could be involved and how important they are. That's certainly true.
Okay, thank you so much, sir, for the answer.
Yeah, the other thing is that we used one method, which is called tDNA activation tagging. And this was described in our Nature publication from 2000. What we did there is we did a functional screen for a transcription factor. So we were looking for implanta for an increased expression of target genes via a certain trick. So we had a certain trick for this. And in this way, we came out also to a transcription factor. And so this was a functional screen. So we knew already, due to the nature of the screen, that the transcription factor was going to be important. So it depends on the type of way you find the transcription factors. If you just look in databases for transcription factors or look at transcription factors which bind to promoter, you will end up with lots of transcription factors, and then you have to sort out what they do or what their importance is. But if you have a screen where you already look for a very specific function, then you know that the transcription factor is going to be important. So the nature of the screen or the way that you find it is very important for, yeah, what you know, essentially.
Okay, how about the answer? Is it clear enough?
Yeah, it's enough. Thank you so much, sir.
Okay, thank you very much. Next question from Joanna Maria. The question is there any way to enhance the role of transcription factor so that they can produce more TIA?
Yeah, there's several ways you can enhance it. So the most simple way is to enhance the expression level of the transcription factor. And that's what we did. So we are simple people, so we did a simple thing. So the disadvantage of the method is that when you overexpress a transcription factor, you might find toxic effects. And that's actually what we found. So some transcription factors, when you overexpress them, this is not compatible with normal growth. So essentially, you cannot find such plants or cell lines. One way to solve this is to overexpress them in an inducible way. So there's various inducible systems. And for example, for the MYC transcription factor, what was done by various groups is to these MYC transcription factor is regulated by the JAS repressor. And actually the JAS repressor binds to the MYC transcription factor and in this way inactivates it. And what various groups did is they made a mutant MYC transcription factor which cannot bind anymore to JAS. So in this way you make this transcription factor independent from these repressors. And this was very successful in making a very active transcription factor. So you can either just overexpress it, and the best way is to do this in an inducible way, or you can modify mutated transcription factor to make it more active.
Yeah, thank you very much for the answer. We're going to the next question. Fatima, maybe you can ask Johan directly. Okay, we have a question here from Intan Nuraini.
Yeah, I see it in the chat box.
Yes. So
Yeah, I'm not completely. For example, if you take a normal cell suspension, our normal cell suspension, and you add to the media tryptophan and secologonin, which is possible because these two are commercially available, nothing happens. So the cell suspension will not make any alkaloids. So it's possible, yeah, if you do not express the correct genes, you will not have the alkaloids. And when you induce the cell suspension with methyl jasmonate at the same time as providing tryptophan and secologonin in the medium, you can actually increase or get production of alkaloids. So it depends on whether the genes are expressed in the tissues where these precursors are present. Okay.
Okay, Intan. What is your opinion about Johan's answer? Is it already clear enough for you, or do you need more information? Do you need more explanation? I think it's enough. I think it's clear enough for her. About next question, Johan?
Yeah, I have a question about, let me see, what are the criteria for a good suspension? Yeah, our criteria are, of course, that they grow well and are relatively, consist of relatively small cell aggregates. So the best suspension, of course, is a kind of uniform cell suspension where you have a single cell type. It's not a cell type that occurs anywhere in the plant, but what you like is a homogeneous cell suspension. And of course, you have a problem if you have a cell suspension that consists of many cell types. Then I would not know what the advantage of the cell suspension would be, because then you can just as well go back to the plant. So for us, a good cell suspension is fast growing and relatively uniform.
Yeah. So that was the criteria for a good cell suspension. And thank you very much for all of your participation and engagement in today's talk. Before we wrap up, does anyone have any final comment or question? No more question or comment? Okay. I would like to thank to our guest speaker once again on their valuable insights, and we hope you found this talk informative and useful. And we look forward to seeing you at our next event. And have a great day, everyone.
Okay. Will there be an exam about this talk?
Yeah.
Really?
Definitely, yes. Yeah. They have to make some kind of a review about it.
Wow. Okay. So many of the stuff is published, so you can also have a look at the papers.
Yes. Thank you very much. Okay. Maybe if you want to ask a question more about this topic, you can send me a message, okay? And I'm going to ask to Johan directly, so he can maybe give an explanation to all of us, especially the one who joined with the metabolomic course. Thank you very much once again, and have a great day for everyone.
Yeah, thank you for the invitation.
Yeah. Thank you very much, Johan.
Thank you.